So this is the final session in which we will try to understand the process of transcription in eukaryotes. So far we've understood the basics of transcription and the process of transcription in prokaryotes. Now in eukaryotes, again we will follow the same scheme of study. We will first of all understand what are the different components that are involved, and then we will study about the different steps involved or the stages of transcription involved. Now let us start with the variety of components that are involved in the process of transcription in eukaryotes. Now the first component that is involved here in the process of transcription in eukaryote are the polymerases. Okay, RNA polymerase, just like in prokaryotes. But here the great difference now is that there are three different type of polymerase enzymes that are required for the synthesis of different types of RNA molecules. Now if you remember, there are three types of RNA molecules: the messenger, the ribosomal, and the transfer RNA. Now three different RNA polymerases are required to produce these three different types. One type of RNA polymerase cannot produce all of them. If you look at prokaryotes, if you recall prokaryotes, there is only one type of RNA polymerase, right? There are not many different types. There is only one type of RNA polymerase which can synthesize all the different types of RNAs that are required by the prokaryotic cell. But here we require three. So let us look slightly deeper into this particular aspect. So RNA polymerase 1, which is here, let me use a highlighter. So we got RNA polymerase 1 here, the function of which is to synthesize ribosomal RNAs only. So its dedication is to synthesize ribosomal RNA, and here we got two ribosomes, right? Its function is to synthesize ribosomal RNA, but it does not synthesize the 5S ribosomal RNA. Now what is this 5S RNA? In order to understand this, let us look at the ribosomes of the two types of major classes of organism. So here we got the ribosomes of prokaryotes, and here we got ribosomes of eukaryotes. Now the ribosome is made up of two parts: the larger unit and the smaller subunit. So we got a large subunit and a small subunit. In prokaryotes also we got a large subunit and small subunit. In eukaryotes, the ribosomes also have the large subunit and the small subunit. However, and also one more thing is that the prokaryotes, eukaryotes both contain proteins plus ribosomal RNA. But now the RNA molecules or the ribosomes in prokaryotes, what type of ribosomes are found in prokaryotes? So in prokaryotes there are three types of ribosomal RNA. One is called the 5S RNA. So the prokaryotic ribosome is made up of 5S RNA, 23S RNA, and 16S RNA. Let us just write them down and then I'll explain what this means. So we got 16S, we got 23S, and you got 5S. Now what is this S? S is known as the Swedberg's unit. And what it tells us about the speed at which something sediments, right? So this is basically a value which tells us how fast or how slow a particular molecule sediments down. So what is important for us to know here, that the prokaryotic ribosome is made up of 5S RNA, 23S, and 16S RNA. The eukaryotic ribosome is made up of 5S RNA, the same as here, and then 5.8S RNA, which is not present in prokaryotes, and 28S RNA, and here we had 23. Here we got 28S RNA, which is again different. Their sedimentation coefficient is different. And eventually we have got the 18S RNA. Here we had 16S, and here we got 18S RNA. So if you see closely, the type of RNA molecules that make up prokaryotes and eukaryotes are different with respect to their sedimentation properties. Anyways, coming back to the point, polymerase 1, RNA pol 1, this is more important to us. RNA polymerase 1, what is its function? It basically synthesizes ribosomal RNA, which means all of these RNAs will be synthesized: 23S, 16S, 18S, 28S, 5.8S. All of these RNAs are synthesized by polymerase 1. All it cannot synthesize, the only ribosomal RNA it cannot synthesize, are these 5S RNA molecules. So this is what was important for us to know. That is why I spoke at such lengths, so that you could understand the function of RNA polymerase 1. The function of RNA polymerase 2 is to make pre-mRNA. Let me just write it down and then I'll explain it to you. What is pre-mRNA? Pre-mRNA is also known as hnRNA. HN stands for heteronuclear RNA. Now what is pre-mRNA or hnRNA? Simply put, these are mature mRNA molecules. These are immature RNA molecules which are then modified to make functional mRNA molecules, right? So what is the function of RNA polymerase 2? The function of RNA polymerase 2 is to make or synthesize mRNA molecules, pre-mRNA. You must specify this, pre-mRNA or hnRNA. Here, hnRNA. That is its only function. What is the function of polymerase 3? It synthesizes 5S RNA. You remember? 5S RNA, polymerase 1 could not synthesize this. It could not transcribe it from the DNA. Who is going to transcribe it from the DNA? Polymerase DNA pol 3. Right? And also DNA polymerase 3 has another important function. It also makes the tRNA. Let us go back and see what am I talking about. So this tRNA is synthesized or basically is transcribed by DNA polymerase 3. Another function of polymerase 3, it also synthesizes something known as the small nucleotide RNA, snRNA. What this does is we will see in just a little while. So this is something that you will have to remember, kind of mug up. But here the most important enzyme for us at the moment is RNA polymerase 2. And why is it important for us? Because this is what will give rise to mRNA and basically hnRNA, and then this will be matured into mRNA, and then this mRNA will form proteins. Right? So that's why this is important to us. So that is the first component that we've spoken about in eukaryotic transcription. Let me just move this aside. Now, what are the other components that are involved? The second component is known as transcription factors. What are these transcription factors? Now, if you recall the prokaryotic transcription process, only two major proteins were involved, the sigma factor and the rho factor. But in case of eukaryotes, large number of proteins are involved. You must bear in mind that eukaryotic transcription process is far more complicated, than the prokaryotic transcription process. Now, what is one more complication here? We are studying that. Transcription factors are another components that are required in eukaryotic transcription. What are transcription factors? Let me show you. So here I have got the image of these three RNA polymerase enzymes. So let us just look at only one of them. Let us look at RNA polymerase 1. So here we got RNA polymerase 1 in orange. This is the RNA polymerase. And if you look around, can you see this TFB, TBF, TAF, TAFID? These are all transcription factors. And if I move to the next image again here, in purple, we have the RNA polymerase 2, and around all of these protein molecules, there are so many of these protein molecules present around. All of these protein molecules here are transcription factors. See, can you see this TF3C? This is a transcription factor, and what we have here in sea green, we have the RNA polymerase 3. So eukaryotes also require for the process of transcription, they also need transcription factors. I hope what transcription factors are clear to you. Transcription factors, what are they needed for is something we'll see in just a little while. Next requirement for transcription in eukaryotes is called the transcription unit. Now, I hope you remember transcription unit. What is a transcription unit? Let us recall again. Transcription unit is a segment of DNA. It is a DNA segment. And what is the speciality of this segment? This segment is flanked or covered on the two sides or guarded on the two sides. On one side it has a promoter. On the other end it has a terminator. And in the middle we have the coding sequence. So such a DNA segment which has a promoter, a terminator on two ends, and in the middle that has a coding segment is known as a transcription unit. Now, what about the transcription unit in case of eukaryotes? Let's just clear up the entire board. I didn't mean to clear these up. Let us just hide them up because they are slightly, these are misleading information, and that's why I'm kind of hiding them. Anyways, so the transcription unit in eukaryotes is slightly different from the prokaryotes. Okay, the transcription unit here has a promoter. All right, so let us say this is the DNA in eukaryotes. So it will have, let's use a blue. So let's use a yellow color. This is the promoter region. Great. It also has an enhancer region. Now this is a new sequence or a segment of DNA. Prokaryotes don't have the enhancer sequences. Enhancer segments can be present upstream, which means they can be present behind the promoter region, or they can be present in front of the promoter region. They can be present anywhere. And the function of these enhancer regions or these enhancer sequence is to make sure that the RNA polymerase binds here perfectly. They are basically an they act as a surety that the RNA polymerase will bind here. All right. So that is their only function. Apart from that, the transcription unit is made up of structural gene. So here we got the structural gene. So this region here is the structural gene. You know, the coding region, same thing, the coding region or the structural gene. And finally, the transcription unit in eukaryotes, they end in a terminator or a terminator region, just like in prokaryotes. So how is the transcription unit of prokaryote and eukaryote different? Only in one particular aspect, which is the presence of enhancer region, right? I mean, broadly speaking, there are many other fine differences that you don't need to understand at your level. Now, what are the other components that are involved in the process of transcription? The last component that is involved are many different types of enzymes. Now, if you recall transcription in prokaryotes, the only two major proteins that were involved were the sigma factor here and the rho factor. Only two major proteins were involved. But here, many proteins have already been involved, which were known as the initiation or transcription factors, which we call the transcription factors. But apart from that, many enzymes are also involved, and we will see what these enzymes do in just a little while. Now, having spoken about the components involved in the process of transcription in eukaryotes, we will proceed further and we'll look at the different steps that are involved in the process of transcription in eukaryotes. What are the three? Again, now in the process of transcription in eukaryotes, there are 5 steps, not 3 but 5. The first step here is initiation. Okay. The second step here is known as the capping, and this is a part of elongation. So the second step here is let me just write it down, elongation, not capping, but elongation is the second step. I have by mistake written it here as capping, but the second step is elongation, and capping is a part of elongation, so there is nothing to bother yourself about. The third step here is termination. The fourth step is something known as the polyadenylation, and the last step involved in the process of transcription in eukaryotes is known as RNA splicing. So you see, this entire process is far more complex than process of transcription in prokaryotes. Now let us look at these steps one by one. Let us first of all look at the diagram here. So what do we have here? We have a strand, we have an mRNA strand, hnRNA being produced. This is hnRNA, and then something known as splicing is taking place, and then we got the mature RNA, and here we got the RNA polymerase, right? And something called as capping and polyadenylation is happening. This is poly-A tail and the cap. And let us look into the details of these particular aspects. So let us look at initiation first. How does this process of RNA formation synthesis start? So first of all, there are certain proteins called transcription factors. So before I start, let me just make let's say this is the promoter region, let us say this is the terminator region. Alright, so let's write it as P, let's write it as T, and somewhere here let us say we've got the enhancer sequence also. We don't need to bother ourselves with that, but let us say we've got an enhancer sequence here, somewhere here, and let's call it E. Let's put it with a little bit of red here. So let's say we have the enhancer sequence here. Now, the first step in initiation is the identification of the promoter site. And what identifies this promoter site are these proteins called transcription factors. So there are these large number of transcription factors which combine with RNA polymerase. Which RNA polymerase? Well, it depends. They can be a part of RNA pol 1, pol 2 or pol 3. Depends on what do they want to make. If you want to make mRNA, if the objective is to make mRNA, then we will have RNA pol 3. Anyways, so the first step in initiation is proteins called transcription factor. They go and associate with RNA polymerase. Let us have a look at the diagram here. So we got RNA polymerase 1 with transcription factors here, transcription factors here with RNA pol 2 is here, transcription factors here with pol 3, right? So the first step is this. So now what do these transcription factors do? Transcription factor and RNA polymerase, let's just draw them with, let's use this red and let's use a block. So let us say this is the complex. Let's say this is RNA polymerase. Let's say this whole thing is the RNA polymerase plus transcription factors. So this complex starts moving on to the DNA strand, and these transcription factors, their job is to identify the promoter sequence. So they identify the promoter region. Once they've identified the promoter region, they tell the RNA polymerase to attach itself firmly to this particular site. So the RNA polymerase is now recruited, and what RNA polymerase does next is it melts the strand. You remember melting? It separates the two DNA strands apart and creates something known as a transcription bubble. So once the transcription bubble is formed, the second stage can be entered. Now once that happens, once this thing happens, and I've cleared it up, and now let's see here. So now once this happens, the RNA polymerase moves forward, unwinds the DNA by its helicase activity, and as it is moving forward, the double-stranded DNA behind it is also closed, becomes double-stranded again due to the activity of RNA polymerase. I would like to bring to your notice that all of these processes require high amount of energies, and the sources of energies are nucleoside triphosphates in the case of transcription. Nucleoside triphosphates, NTPs. You remember, I hope you remember the breaking down of the bonds in nucleotide deoxyribonucleotide triphosphates in case of DNA replication. Anyways, proceeding further. So the process of polymerization takes place, and the mRNA is being synthesized. But now the mRNA which is being synthesized is very different from the regular RNA. Right? So the RNA in case of prokaryotes, let's just go back and look at that particular slide. So here, I think this is, so this is the prokaryotic mRNA here. So this is the prokaryotic mRNA here. And the prokaryotic mRNA, when it comes out, it is absolutely ready for protein synthesis. Rather, what happens is, as this particular, I've actually forgot to mention this, but anyways, we'll just cover it right here. In case of prokaryotes, we'll just take a tiny little, we'll just move away from the topic for just a little moment and revisit prokaryotes. In prokaryotes, the process of transcription and the process of translation are coupled. What do I mean to say by this is, what I mean to say is that both the processes take place side by side. Transcription is the formation of, let us assume, mRNA, and translation is the process of reading that mRNA and forming the proteins. In prokaryotes, these two processes take place together. Let me just magnify and show you what is happening here. So you see, in this particular image, the RNA polymerase is synthesizing mRNA. The mRNA is absolutely ready to work. That is really good about the prokaryotic mRNA. It requires no further modification. It requires no repair. Once it is out, while the process of transcription is happening, the ribosomes also come and join the mRNA. They start reading this fresh mRNA and they start making the proteins. So this is called coupling of transcription and translation in prokaryotes. In prokaryotes, this is how it happens. And what makes this possible? The simplicity of the mRNA strand. The mRNA strand is perfect. The moment it is synthesized, it is perfect. But in case of eukaryotes, it is not so. The mRNA strand, the newly synthesized mRNA strand, the newly synthesized mRNA strand, see, for that matter, even rRNA and tRNA, for that matter, all of these three, but we are going to focus our energies on mRNA for the moment. In case of eukaryotes, the mRNA strand that is synthesized is not perfect. It requires modification. Let me tell you why. I'm going back to prokaryotes again. The prokaryotic mRNA has the bases, and all of these bases, I'm talking about prokaryotes now, okay? Please bear in mind. The prokaryotic mRNA is perfect because all the bases will code for one amino acid each, and all of these amino acids will be connected later on, okay, during the process of translation. But in eukaryotes, the mRNA that is synthesized is a mixture of two segments. Let's magnify and let me show it to you. So the mRNA that is synthesized is a mixture of coding regions. These yellow regions are the coding regions, and this region is a coding region, and these coding regions are interrupted by non-coding regions. So they're basically the DNA in eukaryotes, the gene of eukaryote is made up of codes, no code, code, no code, code, no code. But once the mRNA is formed, mRNA is also formed like that. There is a coding region interrupted by non-coding region, coding region, non-coding region. What I mean to say is this particular region will bring amino acids together. This has no information. This region is basically an interruption. So these non-coding regions are known as introns, and these expressive regions of the RNA, mRNA in eukaryotes are known as exons. Exon stands for expressive regions, introns stands for interrupting regions. So now do you see there is this difficulty? Once the mRNA is formed, can this mRNA in eukaryotes start the process of translation side by side? Translation? Can it happen side by side? No, it cannot happen side by side for two reasons. Number one, if just in case a ribosome is present here, the ribosome will only read this much and then it will be interrupted. It cannot read any further, and then you will not get a perfect protein. So one reason is that the translation cannot happen there. Second reason why translation cannot happen right away is that the process of translation is taking place inside the nucleus, whereas the components required for the synthesis of proteins are present in the cytoplasm. I hope you get this idea. Anyways, so now we're going to move slightly further and we'll try to understand the remaining bit. So now in eukaryotes, the mRNA that is synthesized is called hnRNA, or there was another name for it, pre-mRNA. Now, pre-mRNA or hnRNA is characterized by the presence of coding regions called exons and non-coding regions called introns. So you got exon, intron, exon, intron, exon, intron, so on and so forth. And that is why hnRNA or pre-mRNA, now I hope you understand why is it called pre-mRNA. Pre means before actual mRNA. So these are immature mRNAs. So now this is synthesized. It is being synthesized. Now we come to the third point. Now, and this is we are doing elongation now. We've moved way beyond initiation. We are into elongation. Now while the elongation is happening, we've got exons and introns, and also while this process of elongation is taking place, something unique happens, and that is called 5' capping. The 5' end of the mRNA, you see this is the 5' end of the mRNA. I'm not talking about the DNA, I'm talking about the mRNA. The 5' end of the mRNA is acted upon by certain enzymes, and those enzymes, they add guanine, they add methylated guanine to the 5' cap, which is, so this is basically 7-methyl guanine triphosphates are added. A large number of 7-methyl guanine nucleoside triphosphates are added to the 5' end. A large number of them are added, and they are added enzymatically. Now why are they added? They are added to protect the mRNA. Protect them from what? Now you see this is the end. This is the end of the mRNA, and there are these enzymes called nucleases, in particular exonucleases. So these enzymes, exonucleases, are present inside the nucleus, and what they do is, if they find an open end of the DNA or of the RNA, they will start digesting them from the end. And in order to protect the freshly made hnRNA or pre-mRNA, the end or the 5' end is capped, is made to wear a cap of many numbers of 7-methyl guanosine. Right? So this is called the process of capping, and this process of capping takes place during the elongation stage. So now let us say the elongation is proceeding and happening further and further. Eventually this RNA polymerase, and in this case this is RNA polymerase 2 because we are synthesizing mRNA, we are looking at mRNA. So once this RNA polymerase reaches the end, it reaches the site called the termination site, the terminator region. And what happens at the terminator region is that again there are these proteins, large number of proteins which are associated with RNA polymerase. We call them the transcription factors. And similarly, there are other proteins that are associated with RNA polymerase. These proteins identify the termination region, and then they take this RNA polymerase away from the DNA chain. The DNA chain again gets reannealed, it becomes double-stranded, and the hnRNA is now also freed off. It comes off, and here is it. But now you see the other end is open, and the other end again is acted upon by a different kind of an enzyme. You don't need to know the name, but I'll still tell you. The name of this enzyme is called poly-A polymerase. Why is it called poly-A polymerase? Because at the 3' end now, a large number of adenine bases are added. And this process of adding large number of adenine to the 3' end is called polyadenylation. So polyadenylation takes place at the 3' end. So now the hnRNA here at the 3' end, it has a poly-A tail, a tail with large number of adenines, and this process is called polyadenylation. What causes polyadenylation? The poly-A polymerase enzyme. And the 5' end is already capped by guanosine. The G, the cap, basically the 5' cap, it protects the hnRNA or the RNA from being digested by exonucleases. And what is the function of this poly-A tail? The poly-A tail provides stability. Number one, it makes it stable. Number one, it makes the hnRNA and further the RNA stable. And the second thing that it does is it allows the RNA to go out of the nuclear pores. See, all of these processes are taking place inside the nucleus. So this RNA has to eventually go out of the nucleus, and where will it go out from? It will go out from the nuclear pores. And the nuclear pores, and this is poly-A tail is kind of like the driver which knows the path where to take this to, right? So two functions of poly-A tail: stabilizing the RNA molecule, mRNA molecule, and also to direct it out of the nucleus through the nuclear pores. Now comes the last step, which is RNA splicing. Now RNA is still full of these introns, these interrupting regions. So there are again some proteins and special RNA molecules called snRNA or small nuclear RNA. I know this becomes a little too complicated, but then if you watch these videos once or twice, you will be thorough with these. So there are these ribose RNA molecules called snRNA plus a couple of other proteins. They come together, they make these structures called spliceosomes. Let me show you spliceosomes. So these are spliceosomes. Can you see these structures here? These are actually rolled out introns. So we see, let me just magnify and let me show it to you. So we got these introns, and inside the nucleus we got these RNA molecules coupled with a couple of proteins, and these structures are called spliceosomes. And these spliceosome proteins, they act on these intron regions. What do? They cut these introns, roll them like they have been rolled here, and then they cut them and throw them away. So all the introns are now taken apart and all the exons are connected together. And the hnRNA is finally, or the pre-mRNA is converted into a mature, fully functional mRNA. So I hope you've understood the process of transcription in eukaryotes as well. Thank you.